So now the floor is offered to Caspar Schwiedrzik. I hope I pronounced your name decently well, Caspar. From the Neurocircuits and Cognition Lab at the European Neuroscience Institute in Goettingen and the Perception and Plasticity Group and the German Primate Center in Leibniz Institute for Primate Research. Thank you, Caspar. The floor is yours.
Great. Thank you very much. I hope you can hear me.
Good.
Perfect. Okay. So thanks very much to the organizers for putting together this great and very inspiring seminar, for inviting me to present some of the work that we do in the lab. I think it will tie in quite nicely with what we've just heard from Floris and all the great talks that we had yesterday. So the studies I will present today are aimed at understanding how we use models of the environment and how we use them specifically to predict sensory events. So along the lines of a lot of what we've heard already. And I will put a focus on the interplay of predictions with perception and also action, specifically in the domain of high-level object recognition, namely face recognition. And I will first show some studies in humans and macaque monkeys in which we look at the brain's sensitivity to environmental structure using pupillometry. I will then switch to studies in monkeys that look at the single neuron level and with fMRI, how predictability that arises from temporal structure is translated into an efficient and predictive neural code. And we use the macaque face processing system as a model to study that. And then I will sort of transition back to humans and present you some intracranial recordings from an ongoing study that looks at how complex, if you want, conceptual predictions affect sensory processing. And I hope I can provide you with some insight into the effects of internal models of the environment from the very outset of visual processing all the way up to high-level object selective cortex. Let me start by giving you an example of what I mean by extracting structure and predicting. I think we've just heard this also in Floris' talk that we have this skill to extract patterns or regularities. It's a basic skill. We're very, very good at it. So it's a very straightforward prediction that this traffic light will switch from red to green. And we're inclined to learn such pattern completely automatically. And this is one of the things that allows us to make predictions. So one could maybe say that making predictions is something like a default mode of the brain. Now, equally important is, and some of the discussion yesterday centered on that, is to spot deviations from these predictions. So for example, here in Goettingen, where I'm based, we have a slightly different kind of traffic light. So that switches from red to yellow and then again to yellow. So maybe more important than this particular example, usually deviations or violations signal more important events than the color here. And those can then trigger, for example, attentional reorienting and learning. And importantly, if the deviation from the expectation is large enough, this should also trigger us to update our internal models and make new predictions. In the experiments that I present today, we create models of the environment, obviously in the lab, and we do this from temporal structure in the visual input. And then we look at effects on behavior and on neural coding. So what we usually do is that we present these sort of continuous temporally ordered sequences of faces, and they are arranged such that two specific images always follow each other and form a pair. So what we expect to happen is that subjects learn to associate two images that always occur after each other. And typically in experiments, they learn about 9 of these pairs. Just to preclude the question, we don't do the experiments with the face stimuli that I show here. These are really just for illustration purposes. We use these sort of well-controlled computer-generated faces that allow us to minimize the influence of low-level visual properties on the effects that we're interested in. Okay, so in the first study that I want to present today, we are interested in the question whether the statistical structure that we can acquire through statistical learning can be used to dynamically adjust our motor routines to environmental statistics. And for this, we look at pupil diameter. So why do we do that? So we know that pupil diameter affects acuity and sensitivity of visual processing. So smaller pupils sharpen the image and increase the field depth, while larger pupils allow more light to hit the retina, and that increases the signal to noise ratio in the field of view. So that means, in essence, that depending on how much light is available, certain pupil diameters are more or less optimal to sample information from our surround. And this has been quantified in many beautiful ways in the spatial domain. What we wanted to see is whether the pupillary light reflex, so that is the pupil constriction in response to light, can entrain to higher order temporal regularities during statistical learning, and namely regularities that are not a direct consequence of light variations. And if this was the case, so if there was some sort of cognitive entrainment, if you want, this may serve to still maximize information transmission, which is one of the functions of the pupil. And it would maybe, you could say that it's some sort of sophisticated mechanism of active sensing at the very onset of visual processing based on learned models of the environment. So when we flash the phase stimuli during the statistical learning experiments at a steady temporal rate, then the pupil constricts at the frequency of these images. This is also known as pupil cycling. That's also a technique that is used in clinical practice. So basically the pupil opens and closes. So here you see data from a single human subject when we present the faces in a completely random order. And you see it goes open, close, open, close, open, close, just in the rhythm of the images. So now what happens when the stimuli instead have temporal structure? So when we present them in pairs, and so the base frequency is the same, but there's now a structure included in the presentation. What we find here, and I think you can see this by eye, is that there's an additional sort of cognitive frequency response at half the image frequency, so at the rate of the pairs. And I want to stress that there's nothing in the stimulation itself, so nothing in the light variations that causes these responses, but this reflects that the subjects have acquired the statistics of the stimulus stream. It's a very robust effect. So on the right, you see data from average data from 30 human subjects. These are plotted here with error bars. So you see that this is quite robust. And on the left, you see monkey data. This is now a single monkey. You can see that on the single subject level, on both species, we find that there's the expected entrainment at the image rate. And then there's also entrainment at the pair rate that is specific to the structured condition that is shown here in orange. The blue is the random condition where there's the same images at the same rate, but no pairing. Okay, so we see that the pupil light response can entrain to statistics. Now the question is, does that have any perceptual consequence? Right? So we started from this idea that the pupil sort of acts as a filter. So if we look at the pupillary light response, and this is now the average, we can see that there are two things that differentiate the response from structure, which is on the left, to the response to randomly ordered stimuli. So within a pair, the pupil diameter is comparatively wider in the structured condition, as if the pupillary light response was suppressed. And between pairs, we see that the pupil diameter is smaller in the structured than in the random condition, which is somewhat reminiscent of a segmentation of the input stream. And now to see whether these differences in pupil diameter actually affect perception, we ran an additional experiment where subjects now had to detect a small foveally presented disk that was presented either within or between the pairs in the structured condition, and then at temporally matching time points in the random condition. And we adjust the luminance of these disks depending on the subject's behavior with independent staircases. And interestingly, we find that the required luminance contrast to detect these disks is much lower within pairs in the structured condition than in the random condition. And this is sort of in line with the fact that the pupil is wider during that time in the presence of environmental statistics. So we might say that the visual motor system sort of matches the dilation constriction dynamics of the pupil to the temporal structure of the environment. This involves, as I said, an increase in pupil dilation for the predictable stimuli and a reduction of pupil constriction within a pair. And the result is an overall wider pupil during the presentation of environmentally coherent units. In addition, we find this augmented pupillary light response between the pairs, which is akin to segmentation, active segmentation of the continuous input stream at event boundaries between these coherent units. And together, this resembles an adaptive filter in the time domain, that optimizes signal-to-noise ratio at this now ecologically relevant frequency. Okay, so we have an idea from the pupil signal that environmental statistics are used to unify coherent units, to segment units from each other. And the third potential use of environmental statistics is, of course, prediction. So once statistical structure of the pairs has been acquired, the first image in a pair perfectly predicts the second image in our experiments. And now we can also test whether this, once we have learned this through statistical learning, whether this leads to predictability that in turn has any use. And for that, we do a rapid serial visual presentation task in which the subject need to detect the target that is shown here. And then there's a rapid stream of images after the statistical learning session. And the subjects basically have to, as quickly and accurately as possible, detect the target in the stream. And unbeknownst to them, we manipulate the image that directly precedes the target. So this can either be the trained predictor from the statistical learning session. It can be a foil condition where we swap the images out such that familiar images are shown, but not in the learned pairing. Or we also have a novel condition where we show a completely new image that was not part of the statistical learning session. And then if we look here at accuracy of detection, there's a fairly clear result. So face detection is significantly better in the trained than in the foil or in the novel condition. And we can also look at this in reaction time. There's a similar effect. And just as a side note, the pupil entrainment signal during statistical learning already predicts across subjects how good you're going to be in this RSVP task. This predictability that we see as a result of the statistical learning leads me to the second part of my talk, in which we look further into the role of predictability in object recognition. So object recognition is a core task of the visual system, as you all know. And even in this very core task, it seems that predictions are somewhat necessary to carry it out. So for example, if we see this image, we naturally predict that this is a square in a full circle. Seeing a Pac-Man and a square instead is very surprising, although this is exactly what the retinal input tells us. We do this kind of prediction all the time. So we automatically predict, if you want, that this is a single dog behind a tree and not two halves. So it seems that predictions can play a role here in object recognition, but current computational models of object recognition, although they're very successful, they completely ignore predictions and they really just work their way up from the image. But how about the brain? Is this how the brain does it? So an interesting theoretical framework here that I think was in the background of some of the talks that we heard, but was maybe not put to the fore so much, is predictive coding. So in predictive coding, everything, as the name says, starts with predictions. So instead of sort of passively registering what is out there, the brain actually actively predicts what is going to happen and then checks whether the prediction was correct. So whenever a sensory input arrives, higher cortical areas are thought to send a prediction about what the stimulus could be to lower areas through feedback connections. The lower area computes a prediction error. So that is the difference between the input it receives and the predictions. And the prediction error is sent forward to update predictions. And this computation is then thought to replicate across an entire processing hierarchy. And in fact, predictive coding suggests that making and testing predictions this way is a general principle of cortical computation that does not only explain predictions and perception, but of course also action and cognition and so on and so forth. We in the lab look at the neural implementation of predictions in an animal model, namely in the macaque monkey, and there specifically in the macaque monkey face patch system. This is a quite well understood processing hierarchy in object selective temporal cortex, as you can see here in the superior temporal sulcus. And it's basically a small scale system that consists of a number of brain areas that are each dedicated to the processing of faces, and each of them are packed with face selective cells. And the nodes from this spatially distributed yet tightly, the nodes of this network are spatially distributed in the SDS. They're a few millimeters apart from each other, but they're tightly interconnected. And we know the connectivity, so we have mapped this with fMRI. And we also know feed forward and feedback connections from microstimulation studies and from traces. So what is critical about this system in terms of understanding the implementation of predictability is that each processing stage in the ventral part of the system holds unique and qualitatively distinct representations of faces that differ along 2 dimensions, namely head orientation and identity. And we know from now a host of studies that during feed-forward processing, neurons in the face patch ML, sort of at the entry point of the system, they only respond to very specific views of faces, for example, a profile, and they're not so interested in the facial identity. And so they respond to different identities that have the same head orientation in the same way. In the next patch, that's called AL, neurons respond equally to left and right profile views. This is something that is called mirror symmetry. And it's interesting to think about why we actually need that. But for the purposes of today, we just need to know that this intermediate processing stage exists. And at the top of the hierarchy in the face patch called AM, the neurons actually completely disregard head orientation and instead are very selective to facial identity. So they respond to a particular face and not any other face. So one can say that AM representations are identity selective and view invariant. So we now have this nice face processing system. We know the connectivity, we know the distinct representations, and that puts us into an ideal situation to test theories like predictive coding. So we can investigate whether predictions are sent from higher to lower areas through feedback connections, whether neurons use those predictions to compute prediction errors, and so on and so forth. In the experiments I show today, we use the same statistical learning paradigm that I presented in the beginning of the talk in the training phase. The animals are trained for a couple of weeks in this case, not just a couple of minutes like the human subjects. And again, the idea is that after learning, the first stimulus in a pair predicts the appearance of a specific second stimulus. So for example, Laura here predicts Hardy. And during the testing phase that comes after this extended training phase, we present these predictable pairs in a majority of the trials. And then we again create unexpected conditions, changing the first stimulus. So for example, we change the view, but we preserve the identity. And the idea now is that if the system makes a view invariant prediction, then Laura should still predict Hardy, and we should not see much of a prediction error. If we instead change the identity, but we keep the view, then we can test whether the system makes identity-specific predictions. So in this case, because Buster does not predict Hardy, we should see a prediction error. We identify the location of these face areas with fMRI in awake animals, and then we target in this study the face patch ML, so at the back of the SDS here with electrophysiological recordings. And then we can compare prediction errors in the different prediction violation conditions that we control experimentally. And this is highly diagnostic for where the predictions might come from. So the logic is the following. We know that ML neurons only respond to specific views during feed-forward processing. Now, if predictions are transmitted from higher to lower face areas by feedback, then prediction errors in ML should not reflect the ML feed-forward tuning properties, but they should actually inherit the tuning properties of the cells that generate the prediction. So for example, if predictions originate from AL or AM, which we know are view invariant, then the ML prediction error should also become view invariant. Here on the left, you can see in MRI data where we recorded from. This is showing ML in the 2 monkeys that we work with. And now to identify prediction errors, we compare identical stimuli. This is here the second stimulus, when they're either predicted or when they're not predicted. And this is an example neuron. This is a single cell, and we find that these single cells in ML do signal prediction errors with stronger responses when the same stimulus is not predicted than when it is predicted. And this was back then some of the first single neuron evidence that prediction errors are actually computed in high-level visual cortex. We can look further now at the tuning properties of these prediction errors at the population level, and we look at the 2 axes that we had manipulated, so head orientation and identity. So we find that there are sustained prediction errors throughout the entire response period when person identity and view are unexpected. So this is the green line here compared to the black line, which is when it is expected. And the same thing happens when only identity is violated, but the view is correct. This shows that this face processing system actually tests predictions that have sufficient granularity to differentiate identities of highly similar faces. So this is not a category level prediction, face versus no face or face versus object, but this is face A versus face B prediction. Now, when identity is preserved and only the view prediction is violated, then we also observe a small prediction error early in the response, as you can see here in blue. However, the view violation is different from identity violations late in the response. So you can see that the amplitude sort of decays back to the level elicited by the predicted stimuli. Here's a summary of the early versus late phase activity. So we find practically no prediction error late in the response when the prediction occurs on the basis of an untrained view. And this shows that predictions can generalize across views and are not specific to the trained image. So together, that means that ML neurons signal view invariant and identity-specific prediction errors, although ML neurons are sharply tuned for head orientation and not identity during feed-forward processing. So prediction errors in ML actually revert ML tuning, ML feed-forward tuning, and instead seem to inherit tuning properties of higher level phase areas, AL or AM. But I think that's a question that came up a couple of times now. Where do these predictions actually come from? We have some idea about this, and of course there's also still ongoing work to really figure this out. But we can sort of start again from what we already know about the system. So AL is the phase patch with the strongest feedback connections to ML, and is thought to be the next level in the phase processing hierarchy. Here's some fMRI data from an ongoing project by Tarana Nigam, a PhD student in the lab, and you can see here AL on coronal slices. And here's some evidence that, like ML, AL also computes prediction errors now assessed with bold fMRI in the monkey. So given this, we can think about whether AL could really be the source of the predictions, and we can capitalize on the fact that AL is the only phase patch that exhibits mirror symmetry, so an equal response to left and right profile. So if predictions came from AL, this kind of functional signature should also be evident in ML prediction errors if our hypothesis is correct. So by itself, ML responds only to specific views of the predictor, but in AL, left and right profile views of Laurel are basically indistinguishable and should equally predict harm. And in contrast, if front views are the front views are encoded in different AL neurons and should thus not predict harm. And this is actually what we find in ML. So the prediction errors are larger for non-mirror symmetric predictors than after mirror symmetric predictors. So this is, I think, at least an initial argument that ML tests predictions that originate in AL, the next stage of processing. Okay, so I think that these results provide some evidence that the phase processing system exhibits remarkable potential to acquire entirely new environmental statistics through statistical learning. And even more so, the associations that are formed are not on the level of mere pictures, but they are automatically associated on the level of facial identities. That we can see that in the view invariants that they exhibit. And there's nothing in the training regime that preempts this outcome, which also supports the idea that the phase patch hierarchy, that the computational goal of this entire system is the automatic extraction of facial identity. Our results also support some of the central ideas in predictive coding, in that a lower level area here seems to test predictions about upcoming stimuli, which is through prediction errors, a computationally and at least in some theories also metabolically efficient way of neural coding. And during the sustained response, we find evidence for this view invariant tuning, which suggests that early processing stages are endowed with the capability to signal prediction errors that abstract from physical detail through these feedback processes. And in the next project that I want to present, which is ongoing work, we want to look at the limits of this. So how abstract a prediction can actually be tested in phase processing. And for this, we want to look at conceptual representations that include not only the phase, but also additional information about the person, and look at whether this affects phase processing. So I think we are all aware of the fact that we can easily associate information across formats. So for example, a name with a face. And the neural substrate of this so-called conceptual system has been hypothesized to be so-called concept cells in the hippocampus, which respond equally to pictures, written or spoken names of a person. Of course, also alternative suggestions, maybe with even more evidence, that such abstract amodal information is more widely distributed in cortex, not in the medial temporal lobe, but in temporal cortex, in particular in the anterior temporal lobe. And what we would like to know now is whether predictions formed on the basis of such abstract and conceptual information from outside the face processing system actually affect face processing. Now, one thing is that concept cells have so far not been found in the macaque monkey, at least not in the hippocampus. So we conducted studies using intracranial recordings in human epilepsy patients. And the data I showed today are all recorded together with the clinical neurophysiology unit here at the University Medical Center in Goettingen. For the study, we again use frequency tagging, as Lucia yesterday introduced and as I mentioned during the pupil data, but now we do this on neural activity. So we're looking for a signal that reflects now pair structure of our statistical input streams. So before we had this sort of unimodal, purely visual signals where we could frequency tag the pair structure. And now we look for something that arises cross-modally. And to address higher order conceptual representation, what we do is that we either write the name on the screen in letters, or we play a voice that says the same name, and we associate now this either written or spoken name with a specific face through statistical learning. And then entrainment at the pair frequency would indicate that subjects have learned a name-face association independently of the input format, so written or spoken. And we can contrast this with a condition where there is no statistical association between the names and the faces, but everything else remains the same. Okay, so here already some results. This is, as I said, an ongoing study, so take everything with a grain of salt. So we find entrainment to concepts in the hippocampus. This is a single electrode from one of the subjects. In orange, you see a 1 Hertz peak in the structured condition. And in blue, you see what happens if we present the same stimuli, but without structure. And we speculate that this frequency tag may reflect activation of this concept of the person now, and not merely the image. However, we find such entrainment not only in the hippocampus, but also elsewhere. So here's an example electrode from the anterior fusiform that shows a similarly clear effect. And overall, we find some sites in the medial temporal lobe, which is shown here in green, in the hippocampus and in the amygdala, to be specific, where they have been previously described on the single unit level. But we actually find the vast majority outside the medial temporal lobe in temporal, parietal, frontal, and occipital lobes. And just for the aficionados, about 64% of these sites are left lateralized, but that may also reflect some sampling bias in our population. Okay, this is now an indication which brain regions are involved in acquiring these kinds of abstract models. How about the use of these models? So to address that, we again have a second phase of our experiment where we present the learned pairs and different violation conditions on individual trials. We either play the name acoustically for 250 milliseconds or show its written form for the same amount of time. Then there's a 250 millisecond gap and then a phase for 250 milliseconds. And the phase can again either belong to the name, or we change the head orientation, or we swap the identity. And these are basically the violation conditions that I will focus on today. And that's very similar to what I presented in the prior experiments. We also have a control for novelty where we present the previously unseen phase, but the analysis are still pending, so I will not focus on this. And the subjects have to tell us whether the name and the phase belong together on every trial. And what we're interested in is whether and where there are prediction errors in the brain in response to the phase, irrespective of the format of the predictor. So whether it's visual or auditory. And this is basically a 2x2 design with predictability and modality as factors, and we can then look for the main effect of predictability. Okay, so where do we find such modality-independent prediction errors? We are looking at intracranial evoked responses, so something like between 1 and 30 hertz here. These are responses for predicted versus non-predicted phases, again, irrespective of the modality of the preceding name. And at 500 milliseconds, the phase comes on the screen, and the orange trace shows the unpredicted condition, and the blue trace again shows the predicted condition. And you can see that there are cross-modal predictions that affect processing in ventrotemporal cortex, but also, for example, in parietal cortex, and in sort of the medial temporal cortex, closer to where auditory stimuli may be processed. Overall, again, we find a majority of sites that show cross-modal prediction errors in temporal cortex, where the phase stimuli may be initially processed, but also in other parts of the brain, as shown here. We find these differential responses in all the subjects as the entrainment responses. And overall, this suggests to us that high-level conceptual associations that subjects have learned can sort of reach down all the way down to sensory cortices to serve as abstract predictors for phase processing. And we're now in the process of determining the interactions between the sites showing entrainment to concepts as possible sources of predictions and the sites where they are tested. And we, of course, would also like to assess the causal relationships with intracranial, intercortical stimulation in the future. So this is already my summary. I hope I'm still on time. So our results indicate that the phase processing system is highly malleable and can learn statistics of the environment. It uses this information to make predictions for visual object recognition. Based on the tuning properties of the prediction errors, we can see that predictions are passed hierarchically from higher to lower areas, and this preserves more abstract or invariant representational format of the area where the prediction originates. And this enables the brain to sort of generalize from image-specific predictions to more abstract, if you want, rules. And that sort of reveals deeper knowledge than merely associating two specific images. We've also looked at this in other domains, what else is carried by these associations, for example, reward, and I'm happy to show you some slides on that if you're interested. We see evidence for predictions and their test within the core phase processing system, but we also find that phase processing is penetrable by more conceptual information that stems from cross-modal associations. And finally, the statistical models that we learn from the environment can be used to control motor actions in the spirit of active sampling to optimize transmission of information here through the modulation of a brainstem reflex pathway. And together, I think this exemplifies, as of course all the previous speakers have also told us, the efficiency by which the brain exploits environmental redundancies for neural coding and for behavior. And our ongoing projects are aimed at understanding sort of the dynamic interactions between the different processing stages in the phase processing system, their causality, and their behavioral relevance. So stay tuned for future studies from our lab, and thank you very much for your attention.
Thank you, Caspar. Thanks a lot. So, usual drill, time for question now.
Go ahead.
Check the question and answer. I see that Maria opened up her camera, so maybe she wants to ask something. Go ahead, Maria.
I do have a question, but I see there's attendee questions too.
Okay.
So it's...
Let's get the attendees first then. So, Dajit, you're allowed to talk now.
Hi, Caspar. That was a pretty amazing talk. I was thinking, what do you think would be a mechanism for the invariant, inheritance of the characteristics? Like, for example, when we get to the Thomas's picture, and then we predict that we will be seeing Hardy, like the prediction error that we get when there is a different view or profile of Hardy is like gets suppressed after some point of time if the data like we see is correct. So now do we think that like in a different stimulus where we see some other person, like not Thomas, and we predict someone else, like in both of these cases, like the ML neuron would signal like some like identity prediction error? Like how does this inheritance happen? Like is there like a hypothesis for that?
Yeah, that's of course what we would like to figure out. And with the data we have so far, we cannot conclusively answer this, but we have some ideas or intuitions about how this could work, right? One thing is that then associations probably learned at different stages of the processing hierarchy. And as I mentioned, for the view if you learn something on a view invariant level in association, then you can also not make a prediction about views anymore, because the neurons don't, so to say, know anymore what the view is. So if you're feeding back a prediction like this, then you would potentially feed this back to all the neurons that you have been pooling from in creating this invariant prediction, this invariant representation that now makes the prediction, right? And that is possibly how these sort of very precise predictions come about, right? That you have a feed-forward input from view-tuned neurons to view invariant neurons that then feed back along their to the same neurons from which they are building their representation. And, we don't know that, but this is sort of what we speculate about. So I think, taking like a very simple pooling model with some speculation about the connectivity profile of these neurons allows you to sort of think about at least some very basic coding scheme, how you can sort of explain away some activity in the lower areas based on invariant representations in the higher areas. Then, of course, there's another question, like how do you actually suppress activity given excitatory feedback connections? So do you need an inhibitory neuron in between, and how do they need to be wired up? It's something that we can't address at this point, but at least we can think about it.
Thank you. That was very good. Thank you.
Thanks a lot, Yudhajit, for your question. And then we got Fernando. You can go ahead, Fernando. You can talk.
Hi, thank you. Hey, Caspar, thank you.
Hey, how are you doing?
I'm all right. Thank you for the talk. I mean, it's probably aware. I'm pretty interested in these kind of questions. Now, regarding the general framework, this is like really interesting, and it's very important to understand how these feed-forward and feedback connections are interacting, and potentially, how they can be exploited in behavior. But when I think of this from a kind of coding perspective, one thing that I would really appreciate if you could kind of give more kind of thoughts on, or maybe also a little bit of feeling about the populations of neurons. So we all know the paper from 2010 from Crepaldi, and it is like a really strong tuning, unimodally tuned, like in ML, and it's a very strong kind of mirror symmetric tuning, almost as high like in the preferred peak as in the mirror symmetric peak in in AL. So now we got this predictive coding scheme operating and feeding information back. Because it comes from the most anterior patches, possibly, it has this kind of more invariant nature to view. But then the question becomes, what is the prevalence of these neurons? And how strongly tuned and robust is the tuning of the earlier neurons like ML to view? So overall, the population, how strongly is it affected by the signals? Not just if they exist, but what are...
Okay, I have some, let me share my screen again. Maybe I can answer part of your question at least. So of course we determine the head orientation tuning, identity tuning, and so on and so forth for all the ML sets that we looked at. I can only talk about ML at the moment because we didn't do these recordings yet in AI. Let me see, just a second. Okay, I hope you can see that.
I can.
Okay, so what you see on the left is basically the head orientation tuning without a prediction, right? So this is the head orientation tuning to the first phase in a trial where there is no ability to predict anything. So this is something close to the feed-forward conditions that are typically tested in the system. And you can see that this is relatively, like the average response over 80 neurons. You can see that there's a strong and sustained head orientation tuning, preferred versus non-preferred views, right? And it is sustained throughout the entire response period in this population of neurons. what they were selected for is that they're phase selective, right? They were not selected for the strength of the head orientation tuning in any way. Okay, on the right, you can see the same data again now as the slope over the activity in the sustained response. I don't remember exactly the timing, but let's say something like 200 to 400 milliseconds, something like this. And in black, you can see this again, like in the non-predictable conditions, this is fairly stable. You keep this head orientation tuning, whereas in the predictable conditions, so in response to the second stimulus, in the violation condition, you see that in the violation condition, this goes down significantly. Right. So I think what this indicates is that you somehow need to know as an AL neuron what the spike that you're receiving actually currently means, which is complicated, right? Because it could either be in a non-predictable context, a spike that indicates something about, you know, what image is on the screen and what is the head orientation, or in a predictable condition, it could tell you something about what is not on the screen. And this does not have so much information about head orientation anymore in this space, right? So in principle, you would like to have a population of neurons that basically tells you something like expected, unexpected, expected, unexpected. Let me see whether I have a slide for that question. Yeah. Okay, so within this phase patch, we don't seem to have this population of neurons that just indicates whether things are expected or not. So all the prediction errors occur for stimuli that the cells prefer. So we can independently determine the preferences of the cells. We do the analysis typically on the images that the cell responds to in this independent, you know, measurement for preference. But if we look at the ones that the cell doesn't prefer, there are basically also not many spikes. And importantly, there's no difference between predicted and unpredicted. So these neurons by themselves do not necessarily indicate in what context the spike occurs. But we know from other data in humans from intracranial recordings that there are also general deviance detectors in the brain, and they're intermixed with the sensory representations. It's just that at this level and in this phase patch in this experiment, we haven't found them. But in the intracranial recordings in the auditory system in humans, we can see them. So there are both general deviance detectors that just become active if something unexpected happens. And that could provide basically the context for the receiving population to interpret these spikes that are coming up. I don't know if that answers your question, but that's sort of what we're currently thinking about.
Thank you, Caspar. I think we got time for a quick question. So Maria, you go ahead.
Oh, so my question is refocusing everything to the beginning of your talk, and potentially is easy to answer. So you were showing these really beautiful data about how the pupil entrains to predictability within these image pairs. And then you had this behavioral performance with the disc being more detectable within a pair, but only in the regular condition, not in the random condition. And then you interpreted this, and this is what my question is focused on. You said, oh, that's because the pupil is larger in that interval, and that contributes to better detectability. And so I wonder whether that, you know, what evidence do you have to justify this claim, in that it's possible that this higher detectability is driven by the kind of processes that we discussed yesterday. It has to do with there being a high predictability within that, you know, in between these pairs. It means you have less resources that you have to devote to the upcoming stimulus, and hence you can better detect this disc that comes in between. So what is the link between, or is there a link between the detectability of the disc and the pupil size beyond the circumstantial evidence that they co-occur?
My claim was not, it's a little bit more specific what I'm trying to say here. So I'm not saying that they're more detectable because they're predictable. I'm saying that the disc is more detectable because the pupil is wider within a pair than between pairs in the structured condition than the random condition. So yes, why is it wider?
Well, I know that it's wider, but I mean, why are, so, and because another interpretation for the increased performance could be the at that point in time, sensory information is the, you know, the, so to speak, the sensory information, not the disc, but everything else is more predictable, which means that, they're in a state where they are using up fewer resources, they know exactly what's coming next, and so on. And so they have more resources of whatever nature to allocate to detecting the disc. So the explanation is not necessarily because the pupil is larger and hence the disc is more physically visible.
Yeah, so it's possible, right? So that I think that it could be that resources are freed within this unit. That is a possibility. It's also possible that you somehow follow attentionally those units of pairs, yeah? And that you have an attentional rhythm that follows this frequency, right? So I cannot rule this out based on data. I'm not entirely sure that I need to invoke attention or resources here, because it's like an additional consideration beyond the widening of the pupil that we know lets more light through, and sort of simple explanation for why you could see something more easily. And the occurrence of the disk itself is not predictable, right? So the only thing that is predictable in a sense is the second image in a pair if you've seen the first one. So, but
I guess you could run another experiment where instead of the disks, you use faint tones. And if you show the same pattern of performance, where the faint tones are more detectable when they're between images than across, then that would suggest,
Yeah, absolutely. We have that on the menu for future studies. Yeah.
Oh, cool.
It's an interesting question whether it's an overall, like also whether the pupil reflects something specifically about visual processing, or it's a reflection of a general system that is acting. Right? There are some dissociations based on older studies in patients that suggest that pupil entrainment, or this pupil cycling effect that we see here, is not a reflection of this locus ceruleus story, but relies on different system. So I would not, also in terms of the latency of these effects and so on, not throw this in one box with the overall arousal story. But of course we haven't shown that, and the data on which I base the statement is really little.
Thank you.
Thank you. Thanks a lot, Maria and Caspar.